G'day, good morning, good evening. I hope you're well where you listen. This is a medical cannabis show and I respect the law and I hope you do too. Today we're not going to discuss anything really prepared. The only thing I could talk about is it's been World Compost Day or World Compost Week, something like that. So if there's no real questions, I can elaborate on World Compost Day, or is it World Compost Week? Every May 29th, apparently it's Composting Day. So that was last week. So yeah, that's about the only thing I really can think to talk about. But I hope that you cool viewers will have something else to discuss and I can help with your questions a bit better. So I'll go and wait till the more normal 2-minute mark for people to arrive. So fast forward if need be. You can just wait. Get that background music. And then yeah, you probably get a copyright strike. Just got to be careful. 30 seconds and then I'm just going to start talking about compost, I think. World Compost Day was 5 days ago. This looks like what's going to be discussed. Apparently there's a few people here. Not much has been said in chat yet. Please drop your questions and chat suggestions and stuff like that. All right, that's been enough. So I'm going to talk about compost. Really? That's what we're going to talk about? See, I like to help people with their topics because otherwise it's my chosen topic and I don't know if everyone wants that. So I'm going to briefly talk about it, yeah, just to get me to the 10-minute mark. And if there's been no questions, it'll just be a short show. I think that's a good idea. Right, there's the plan. Good organisation. So let's start and share, and I'll show you what's going on here with World Compost Day. Every May 29th, learn about Composting Day. So that's what I can discuss. Oh yeah, here's some cool questions I was reading down. Is human urine a good compost activator? Oh, well, what is compost? It's a bunch of breaking down material. Is a good way to put it. So you need heaps of microbes for that to start, and you need heaps of nitrogen or ammonium for those to feed your microbes. So yes, urine will help, but how much? Can you put too much in? Yes, you can, and you can put the wrong urine in. If you've got antibiotics in your body, they're going to be peed out as well. So you have to watch the source of all your inputs for your compost to make sure it's going to be organically safe and the process is going to happen correctly. What other questions? What is compost? Oh, this was just, yeah, breaks down organic matter. Breaking down of organic matter. It's so nutritiously active and it holds just so much water too. How often do you water a compost pile? Oh yeah, that is pretty cool. Well, you want it to be 60 to 80% humidity. So, and cover it if possible. You just want the moisture for the microbes to start activating, which is about at least 80 for bacteria. It's minimum 60 65 for some yeast. What is the first rule of composting? I don't know. Is blood and bone good for compost? Yes, it is. Are gum leaves good for composting? Yes. You have to watch the pH. It should neutralize, though, if it goes through its full cycle. What happens if you don't turn compost? It can become anaerobic. That just allows the oxygen back in. But also you can do an anaerobic compost. So it just takes a heaps longer, because the oxygen, which is an electron acceptor, when it's the best one, it's more readily available when oxygen's around. So that's why, otherwise with the microbes, they've got to get their oxygen out of things like that has already absorbed nitrogen, like nitrate, NO3, and sulfate, SO4, stuff like that. So that's why it takes longer with anaerobic composting. Does compost need a lid? Well, you need moisture retention. So whatever you need to do, the 60 to 80%, to keep that up so it functions well, and the right ratio too. Oh, look, Jono Who Cares is in. Maybe we might be talking about something. Hello, Jono Who Cares, and everybody else listening. I couldn't think of anything to talk about today, so being World Compost Day a few days ago, I thought I'd just go through a few little hints and tips I picked up for rad compost for you to get a good harvest of it. So ask some questions if you want. Otherwise, I'm going to talk about composting for another few more minutes, and then that will be it. It'll get me to the 10-minute mark. So I suppose, all right, you just want to see some. There's nothing worse than people just saying, well, I prefer seeing slides, because you don't know if that person has up-to-date information, or if they just had a rad smoke and all of a sudden they've lapsed a few words. So you can't beat the slides, I reckon. What is it, composting? This is in soil. These are my soil slides. We should go to some study where organic farming, this is a better organic thing that I've done. There's no module section. Yes, it is down here. Oh, peanuts. I just got some peanuts. They nitrogen fixing, promote nitrogen fixing bacteria. Organic concept. Organic food, no. Vermicompost. Which is this one? Should just go to my slides actually. Oh, wrong button. At the top here, pictures. Lecture 3. Lysometer, that's right at the end, lecture 10. Oh, there's the start. It says lecture 10 start. Oh, the start. So the end must be started. It must be down here. Lecture 40. 9th of the 13th. 8th. Oh, so this is the start. All right. Nice. I'd like to do living soils farming compost. So says Jono Who Cares. All right. Let's elaborate on some compost stuff. Water requirements. Oh, gee whiz. Here we go. So, properties of good quality compost. So this is when it's harvested. Because look at the percent. So 25 to 30, that's what you store it at. So afterwards, after it's gone through the process, you want it to smell pleasant and an earthy smell. The name of that smell's geosmin. Geosmin. And you'll learn that and you just know there's no other funk to it. It just smells like earth. And that's that name of the terpene that you get out of the smelling after it's been done properly. And see the pH, it's neutral. It's up around the 7. So it's rad. Everything stabilizes. Carbon and nitrogen ratio. So the first one's carbon, and then the second one, so you want is a 25 is to 1. So 25 carbon to 1 nitrogen, because otherwise it's too high in nitrogen, it will use up the nitrogen and not store it in your compost. That's the temperature, because you want to maintain, it'll go into the, this is storage temperatures. It'll go into the thermophilic range there. That's the mesophilic. And above 45 is thermophilic, just to kill all the seeds and stuff like that and the bad pathogens. That's why compost is rad. Oh, look, there's Lene. Good morning, Lene. How are you? A very good humic content. And look at the nitrogen, because it's like you've got your ratio right. And earthworms, you can actually get the wrong sort of earthworm, believe it or not. There's 3 types of earthworms. And actually, there's a chart up here. All right, I'll just enoic, which is the real deep ones, which is the large ones, and they actually eat worm castings. And then the top 2 is the better ones, epigeoic and epigeoic. See, can't remember now. But you want the top 2. Oh, look, it says it actually over here. Oh, endo. Okay, epigeoic means on the top, and then endo is just underneath the top. And they're the 2 species that produce worm castings. The other big, massive ones, so you've seen that it's huge. See the size difference? 8 centimeters. These only get to 12. So the sizes go with the smaller ones. And the outside of the worms actually secrete ammonium, which is one of the readily available plant nitrogen sources, one of the 2 types it accepts. One of my teachers was a worm expert. Waste characteristics, animal manures. Oh, here's the different ratios, because how do you know what sort of ratio I've got? So it's very wet with all this sort of stuff. It's got a low carbon to nitrogen ratio, but if it's got some dry stuff in there, biomass, it's a little bit higher and better. And then the highest, so if you've got this bottom one, you can just add some sawdust, and then it'll come down, bring it back to what the level should be. These courses go into extreme depth, and I'm not going to go into them. There's some layouts of some beds. Here's some cool ways to do it if you don't want to do vermicompost. Vermi just means worms, so it's just adding just worms to your blend. So it's pretty cool. It's just bamboo and everything. Look at this one, a multi-tiered vermicompost bed. Look at the size of that thing. And that's done with all wood in the jungle. I take my hat off to people like that. That's really well done. That's going to handle storms and all sorts. So any budget you can do it. Worm preparation. I suppose, well, this is a compost method. It's just using worms. So vermicompost accelerators. You can add these microbes and it'll actually improve the cycling. Nutrient cycling, and that it'll happen a lot quicker. That's the end of the slides. Oh no. Oh look, here we go. There's the 60 to 80%. So in the composting actual process, that's the moisture you want. And how do you know it's that? If you haven't got a gauge, you pick it up, and when you squash it together, you just want a couple of drips to come out. If heaps come out, it's too wet, and if not, and nothing comes out, it's too dry. And how to harvest vermicastings. Yeah, it's pretty hard, surprisingly, just to get the actual black goodness, not the worms with any worms in it. So you can make these high towers, and all the worms actually go to the bottom. So if you make little pyramids around, scoop off the top, make them again, scoop off the top, and keep going around. That's a way. Or you can just go through, put down mats like I used to, and just go through it all. Make pyramids, scoop off the top, and then yeah, it's what's left at the bottom. Works pretty good though. You put all this effort in by doing this, and that's all your minerals and vitamins for your grow. So people can either do all this effort at the start, or they can just feed it from a bottle. So it really pays off. Advantages of organic farming. It improves the soil health and quality, disease and pest resistance to plants. See that? So that's one thing that you don't get from synthetic growing, natural disease and pest resistance, because of all the other pests and processes that happens that engages different pathways in the plant. So it can develop these genes that are primed and ready. And also it gives you a better secondary metabolite profile that you won't get from synthetics unless you're adding salicylic acid and all these different other types of things to boost it up and manipulate it. And another advantage of organic farming is recycling agricultural waste, lower input costs, and the environment is just going to full on win. It's all about sustainability. Why enrich your vermicastings? I'll say good day to Lene and whoever else has arrived. There you go, Lene, good morning. Oh, Blinky's here. Hello, Blinky. Worms rule. Yes, they do. They're actually being called the intestines of the earth. John, who cares? Just a quick question. The microbes, how and where does one get them? I'll go into that. Good question, Jono. It's from, cut a long story short, it's from poo, manure. So they are like, there's billions of different types in the manure. So that's a good way of starting it. So you have to find out what inputs go into that animal that you're trying to output. Like, for instance, a kangaroo, you know, their manure is sweet unless they got diseases or something, which they don't really have. So, and a bad one would be horses, because they get shot with all these different inputs with antibiotics and steroids and all these things, which is going to leach out into your waste. So that's why you've got to really, truly watch where you get it from. Bokashi is fermented grain. So they've just got this grain that they've soaked microbes in, and then it's inoculated into the grain. So that's another source that you can get. Bokashi is quite cheap. Yeah, your manures, your bokashi. You'll get some from your green vegetables and your leafy vegetables. There'll be all sorts, anything's got it on it. So all the stuff that you're going to, the variety that you're going to put it in, of all your sources should have microbes and fungi, all sorts all over the top of them too. But to increase them, you want to get some beneficials. So you'll find out what beneficials is good for medical cannabis, for instance, and then you'll go and inoculate it with that too. So that's another good thing you can do. We'll get to that though, because it'll come through that second. Microbes rule, mate. They're so important. Blinky says, my 3 jars of recent bud are beyond my expectation and very happy. And a very happy cat. That's the way. Very good, Blinky. Hello, Genetic Memory Farms. Nice to see you, mate. Compost is our most practice source of microbes. Could be a manure or could be a decomposted plant material. Oh, yeah, you're just helping with Jono's question. Jono Who Cares question. Good on you, mate. Thank you. All right, we'll get back to sharing. See what's wrong button. Sorry about that. There we go, entire screen. Now, why enrich vermicastings? So why would you want to enrich them? So that's kind of what you're talking about, Jono. And for different grows too, you'll want to enrich them. I've made a video on enriched vermicastings and how to do it, and the experiments that were done, and you can see the results from the tests that I went through and did, test result sheets. But for this slide, it says why enrich them? Because the NPK and minerals that are in your vermicompost can only be so high. So if you want to, I've got a nitrogen pig for a crop or a phosphorus pig for a pink death bubble or something, you might want to just go and boost these up a bit. And yeah, that's the long story short. There's heaps of slides, so I'm going to get through it. Nutrient enrichment strategies from natural resources. So if you've got your nitrogen pig, how are you going to add more nitrogen organically? These are some of the good ways you can do with all your cakes. Neem cake is something that's got beneficials in it too. Might heap some microbes. Poultry waste is good for nitrogen, but you have to be careful, you know, putting too much in. And leguminous crops like that red clover or the peanuts, stuff like that. What was I going to say? Poultry manure. Poultry manure is good too because it adds a little bit of alkalinity to your substrate, And it's very hard because most processes always make it more acidic, adding the proton, the hydrogen plus into the substrates. So that's always beneficial too. Saves you. So people might use lime as an example. Rock phosphate, bone meal for phosphorus. Look at the percentage it's got in it, it's high. So if you think, and this way too, it will stay on the ground. You'll recycle in your pot. You'll recycle it, hopefully. So after you've finished it, you'll tip it out. I've made a video too on root tea, how to get the microbes out of your roots and to how to enhance them, how to enhance the numbers in them, which works really well. And I'll show you how and the numbers on my gauges, how it goes up. But right back to it. So these are just some natural ways. So you come back to this slide if you want and see how to boost up your vermicastings or your substrate that you're trying to blend and make. Preparation of enhanced vermicastings. So they, in this slide, they use cow dung plus aquatic weeds and agricultural wastes. So that's it. Then they add that to the earthworms or vermicompost, and then that's the end result. And then they've gone over here on the left-hand side, and they've also put in enhanced it with microbes. So they've got some beneficials that are very useful in their environment for their grow, and put them in. Like azotobacter. Azos are really good nitrogen fixing. So they just, and they're non-symbiotic. So they'll work with any crop. So what they do is they pull nitrogen out of the atmosphere and they use oxygen and convert it into a nitrogen source that's useful for plants. It'll be like nitrate, or it'll be nitrite or something that'll need to be broken down again, but it's very handled to get nitrogen in your substrate for free. This other one's Bacillus polymyxa. That's phosphorus solubilizing bacteria. So you've got to have your phosphorus in there for that to actually solubilize it. It's different from, and another azospirillum. That's really good for the fungus. That'll do on this one. So you just do it, yeah, that way. It's so rad. Here's the different examples of what it comes out like by adding lime, dolomite, mica. Yes, the next one. This is the experiment. I'm not going to go through them. Take-home messages. This is a good down the bottom. The effective shelf life of vermicastings in storage is for a period of 90 to 105 days. Storage for longer periods, like above 105 days, reduces the microbial activity and the nutrient availability of the vermicastings. So it's something to be pretty aware of. It's not all good, and especially the tea. If you get vermicastings tea or leachette that people have taken out of the bottom of it, which is so nutrient rich, like so microbially enhanced and nutrient rich, you have to dilute so much. If you put your ppm meter into it, it doesn't work because it's way too high for me anyway. So I've got to really dilute it. But that's right, it's shelf life. It's only 3 or 4 days for the teas. So for people to sell the teas that you see in the shops or whatever, or at markets, ask them how long it's been in there. If it's been more than a few of those days, oxygen's been depleted in it because it has so much microbes in it, and therefore the anaerobic ones are starting to enhance. So you might be poisoning, or not poisoning, you might be putting pathogens into your substrates. So be careful about the teas. Precautions regarding the use of plant extracts. No. Quality criteria for vermicastings. Yeah, because it's a compost. We're talking about that. Physical characteristics. There's 3 things to soil. There's the physical, biological, and the chemical. So you want to make sure all those characteristics are high, and it does. It enhances them all. Vermicompost is rad. I've used it for many years. Quality criteria. So you can go through here and see how much yours has, and it should be equivalent if it's gone through after you've harvested and it's broken down. Oh, yeah. GMF says he's using water from boiling potatoes, beans, pasta, Rinsed yogurt containers, etc. from the kitchen that I don't salt, and it adds kitchen scraps to the worm bins. Good work, mate. I'm in LED land. Okay. GMF said that let the kitchen water grow some microbes, then add the water to the bin. Oh yeah, yeah. Because microbes are everywhere, Jono. You asked the source of them before. They are in the air, and all the airborne ones are mostly healthy for us. That's why they're in the air. We don't get sick from them. It's all of the anaerobic ones that we get sick from. I love talking about microbes. I haven't talked about them for ages. I want to talk about microbes. How do you get them? You can get them out of the... All right, let's go and talk about microbes. Typical signs of pest and disease indicators. Yes, if you don't have a good diversity of microbes to take care of all the problems, there's a high chance that you will get problems. That's why living soil is so buffering. In that way, it takes care of itself. All right, let's talk about microbes. Biopesticides. Yes, just some of this too. Need the words bios, living pesticides. So you go and add beneficials to your problems and it takes care of things. And it really, really does. The 3 levels. Oh, let's talk about microbes. I'm going to find a microbe section. Microbes are fun. Oh, yeah. Can you do oxidation on the worm tea? That's how you make it, Jono. You put bubbles into it. Watch some of my videos. It's easiest to explain because I really go into detail and show the slides on it. That's, yeah, just add bubbles. You need to, it's cell culture. So you want to multiply cells. So you need a few things for that to happen. Yes, a carbon source, oxygen source, electron donor acceptor. So there's a few things like that you need. Oh, sorry, Blinky says they have a live aloe plant living in a deco vase. My plants love water. That's the way. Aloe is very useful. Oh, yeah, GMF. Try to make low waste. Very good. I need aloe. All right, let's show some microbe stuff. My voice is getting a bit sore for some weird reason. Microbes. How to get them. Beneficials. I've done 4 microbe courses at university so far. Pesticides. Oh, here we go. Microbes. If you get the correct microbe, it can treat all of these conditions, believe it or not. Oh, white pillary mildew. Well, it can be there. They can take up the spaces so that, like Bacillus subtilis, for instance, it'll be sitting, you'll spray it on the plant, I use that one, and it'll sit there and it'll take up its location. So when the powdery mildew comes into its right, and it's always sitting there, but when it comes into its spot to germinate, it'll be overpowered and Bacillus subtilis might interact with it. It mightn't allow space for it. It might put out a biofilm that it doesn't like, and it just helps. That's an example. Biopesticides against various pests. Oh, that's what I was just sort of talking about. So all of these can be fixed from different biopesticides. So, and that's all organics, just working well to, and if you know your, what's a good example everyone gets? Mold. Right, it was a mold one over here, being fungus. A trichoderma is a good one. That's always sitting there. Trichoderma, yes. Bacteria that work against that as well. Some Pseudomonas species do. And how do you get these? You're saying, look at all these things. Well, you look up beneficials, and then it'll come up under shopping, and then you'll see it'll have different ones inside of it. Like one pack might have azos, for instance. What's that called? Oh, I can't remember. There's only azospirillum in this one pack, azos, and you get your micas, and different ones only have one species in it, but you get ones which have multiple in them. So go through them, like great white shark and other things like that. They've got multiples of these in it. So then you'll culture them up. So you might put a little bit of that into a tea or a brew and bubble it up. Look at my videos. I've made a few on this. And then once you've increased the population of them, because you've given them in their favorable conditions with the elements that they require for growth and metabolism, you can go and add them to your substrates through, you can foliar, spray them, or you can put it under the soil or under your rhizosphere as well. Common garden pests. A good one that takes care of all this is Monterensis. Takes care of a lot of problems, and lacewings. So if you get those two in your area, there's a high chance they will take care of a lot of problems just on their own. And how do you get them? You go to your Bugs for Bugs person in your area, and you pay them maybe $50 each for those two, and they'll send them to you. Then you release them. I've done it a few times. It's pretty fun. This is how microbes communicate, through quorum sensing. And you know how they put that biofilm out? You'll see that slimy stuff around different things. That's their film they've put out. And they've put different things in that to ward off other microbes from trying to invade their territory. And it's got different, what do you call it? Different elements inside of it. It's pretty cool. Some biofilms are good. Yeah, some aren't. I don't want to go into detail. Living Soil Symphony. So if on the right-hand side, it shows it's not living soil. They've gone and used pesticides and synthetic fertilizer, and that's how it ends up. Because that environment relies on their synthetics to survive. We're on the left, which is how Mother Nature's actually got to this date, or sorry, before 150 odd years ago. It cycles well, or the nutrient cycle. So there's so much microbes here, they break down everything, which cycles all the nutrients into beneficial elements. That's leached out and transmitted around different areas, so and it's being refed. So there's just a massive, humongous difference. And what most farmers still want to go this route because it's easier and less labor intensive. So they just pay. But that's what it does. Oh, yeah, you can fix all these things. What's this? Yes. Pseudomonas petita. Oof. Evidence suggests that these foil bacteria work as a biostimulant. Okay. I use those bottom two, not the top two. Compiling the hidden heroes in plant health. Your phylosphere, which is your green bits, your endosphere, which is inside your plants, and then the rhizosphere, which is right next to the roots. So all those areas have hundreds of thousands of microbes on it. And actually, there's a chart that I'll bring up, slide that I'll bring up soon, that shows all the different bacteria and fungi on different parts of the cannabis plant. You see different areas around the world occupy different microbes and fungi from the different activities that goes on in their environment, like moisture, humidity, all the forest and the temperatures. This is pretty cool. You don't see, excuse me, you don't see this very often. It's a pH chart with bacteria and fungi inside of it. So you can see as the soil gets below about 5.5, bacteria start and dwindle. They don't like those conditions, as acidic conditions. So there is acidophiles and stuff like that are there, but there's a very low amount. So that's also a problem. Some hydroponics people and stuff, you know, they like to go down to this low, which has different blockout potentials. Look at the CalMag. So that's why people always say, just add CalMag, because they run lower pH instead of bringing it up a bit, and it would be more accessible. Yeah, that's pretty cool. I like that one. Still to this day, that's not really, I still really like that one. Availability. Going through a bit of cycling. I suppose it's this carbon to nitrogen ratio. How to maintain a carbon to nitrogen and phosphorus ratio is generally sufficient to ensure unrestricted growth in aquifers. Awesome. That was an environmental course. What's this one? Nanoplastics move up the food chain. So if you've got sources that you're putting in your mulch that could contain nanoplastics, you're not going to see it with your eyes, but it's going to be so micro and small like this. It will go in, and then plants including cannabis being a bioaccumulant, it's going to get absorbed into the plant tissue. So if you were potentially to eat that plant, there's many ways you could absorb it into your body. And if you're eating it, you've got microplastics too. So really be careful on the sources of your composting. That's how you test the microbes. So if you want to test, if you get a package that says it's got 1 times 10 to the power of 6 amount, you go, right, how do I know if this is true? All right, you just put it into a serial dilution off this slide. So you'll put an amount in 10 mils in a thing, then you take out 9 mils, and then you keep doing it, and take a swab out there, chink swab, and do the swab on the Petri dish. Do the same. 9 mils, that, and it keeps going right to the end till you can get to a number where you can count it. And you can count 1, 2, 3, 4 times your dilution ratio, and it'll give you the times 10 to the power of, this is 5 here. So then you'll know that they have 4 times 10 to the power of whatever this is test for. So you can test your stuff that you're buying if it's in its favourable conditions. Endophytes inside seeds. Microbes. Well, there's another source, Jono. You can get microbes out of organic growers that produce seeds. Because if you grow with synthetics, they will have a very low endophytes inside of their plant tissue because they want to keep it all sterile and stuff like that because they spray it so much. What's the ideal medium growing of potassium? Okay, why roots grow in excess when phosphorus is limited? We're not talking about elements. Microbes at the moment. What's this test for? Strains and plasmids. There's your Petri swab. So you see how they do the swab there? So you do heaps at the start, then you do a little bit lefts, and then singles. And it should dilute out of the, because you can't count them. Cool protozoa pointers for Primo MedCanna. Oh, very good. Protozoa is the favourite food of earthworms. I like that. Isn't that pretty funky? From the bacteria, if you put it under the microscope, it looks something similar to this, and it really just goes off the shapes. So the coccus are round, the bacillus are like that, like a grain of rice. What else is there? I haven't done this for a while. Where's the spirochetes? Are the spiral ones? They look like a corkscrew. Yeah, that's about the best bit, is you identify it by that. Pseudomonas is pretty similar to bacillus. Enterobacter is round. They're a coccus type of a round thing. Oh gosh, here we go. I'm glad what I was saying is good. There you go. That's one of my little tests. Testing that, see if it's right. Then you put it under the microscope and you can see those shapes, and you'll know, all right, that's a bacillus I'm looking at, or a spirochetes, something. Fusarium, the long looking, you'll see them cruising around lots. Not fusarium, sorry, fusiform. Yes, fusarium's heaps different. That's bad. That's Umycetes. Western flower thrips. If you get thrips, I've made a rad video on it, and you can easily get rid of them, believe it or not, through the biopesticides. So you're going to order your cucumis or hypoaspis, but it's cucumis, I remember, was really rad. Oh, here we go. That's where I actually ordered from. So on the left, thrips. Cucumis works really well, but it's not cheap. I think it's $150 for a container that they send you. Hypoaspis for the soil doesn't do much, but Monterensis is really good. And remember I said before, lacewings. Look, lacewings eat everything. So between Monterensis, here, does all them, all the mites and white flies. Lacewings does the rest. So between those two things, if you're coming into spring, consider ordering those biopesticides. This is cool. Just the different ways, nutrient cycling of a microbial cell. So how does it cycle? You can do all these around the outside. You can accumulate. You can absorb them, attach to them, reduce, oxidize them, which is similar to reduction, mineralize them, leach them out, and bio-oxidize. So all these different processes can happen with one microbial cell. And if it's not in its favorable conditions, like it's right moisture or right temperature, it can't start these processes. So it sits there as a little, like the microbe cell, so it's a grain of rice. Half of that is a endospore. So when it dies off, it'll be left in its form to germinate again. And that's why you get all of these, Jono, who cares? You get a lot of the dung and your poo, different types of manures have these endospores in them. So it mightn't be active, it might be sitting there even dry. And you're thinking, look, it's dry as, but there's so many spores inside of it. When they give them their right moisture, favorable conditions, they're going to come to life again. So that's how you start that up. And John, who cares while I'm onto that, another thing, I hope you're listening, another way to test your microbes in your substrates is the CO2, because they've got to respire. So you can test it by, say if you make your substrates up, or if you've got tents, it's easy, just put a CO2 meter in and you can see it. But that might be with plants also. So to test substrates properly, you can wrap a bag around it and put a CO2 meter inside, and you'll see pretty quickly that the CO2 will just keep jumping up, and that means it's very active inside. Availability. Contaminants can serve as electron donors or acceptors. Yes, that's why you've got to watch what goes into your inputs. Because if you've got all these different types of metals and problematic hydrocarbons, things can attach to it too, and they'll just go through the cycle. And the plant will store them. It doesn't really use them, but it stores them inside of the plant tissue. So that's how it gets cycled around through nature. So if you go and eat your medical cannabis, you can go and eat all these things that's bad, and you can get poisoned from it. So that's why when doctors say you probably got poisoned, yeah, it probably was right because of the street weed that it was. Had all these different substrate problems. Enrichment cultures. If you want to pull, you can pull microbes out of the air. Some of those azos, for instance, if you want to make your own, you can. You have to use aerobic respiration. So there's probably a chart up here. Explains it a lot better because it goes into great detail. I'll just wait for it. We went through that most probable number, that's serial dilution before, so you can count your microbes. Let's see how they keep going, and there's no growth right up here. So you know, all right, it's only times 4. And if you're really into microbes, Brock's biology is insanely awesome. Most of my slides from all these classes come from Brock's biology. I got I think it's the 13th edition now. You can get it online. Isolation of anaerobic bacteria. Very good. That's just trying to identify. So see, they've diluted there, diluted to here. And you can see it's a lot easier to count them. Here you go. What I was just saying before, can you pull some microbes out of the air? You can, but it's only one type, and you want thousands of different types and you grow. But if you want free azotobacter, here's the way how to do it. So you'd separate it. You need 4 Petri dishes and 3 beakers. So you're going for mineral salt medium, mannitol, but lacking ammonium, nitrate, or organic nitrogen. So there's no nitrogen in here at all. So you'll go and put it in down the bottom, the nitrogen. So then you'll plate it so you'll know that that's where it is. You've got it there. Then up the top here, it hasn't got any on it. So you'll plate it and you want to identify the same. Then you'll know that that's the similar, because azotobacter breaks down NH4. So that's where you're trying to get that from. So you can get your azos out of the air by doing it that way. I've never done it. I'd rather pay isolation of a pure culture. That's pretty similar to what we were just talking about. Pure cultures have just one microbe in it. Microbial community from environmental systems. What to analyze? All right, that's a bit detailed. 6 categories of culturability for microorganisms in nature. So that's why 99% of the microbes in the world haven't been named or identified yet, because they're unculturable. They might be symbiotic. They might be living in mediums that is hard to replicate. Many different reasons. But it's so fascinating, microbes. Oh yeah, here's the culturability of them. So you can see in seawater, they have the best they can get to is 0.1% of the whole microbes of the seawater. That is not much. There's an ad on the screen. Oh wow. Okay. So you can see here the culturability of different substrates. So in seawater, the best they can do is 0.1%. Freshwater, where it comes right down to active sludge, because they've done a lot of concentrations in sewage treatment plants and stuff like that. And they've cultured quite a few, 15% of all the microbes that could be possibly there. So it's quite high. But in a general thing, it's quite low, isn't it? Look at these numbers. Preparation of soil dilution. Okay. That's if you want to put it on your slide. So if you want to view them under your microscope, you'll go about it doing it this way. Here's your metabolism that's in microbes as well. So they do have a catabolism to make the energy and an anabolism to use the energy in microbes as well. So it depends where the sources they're coming from. So remember I was saying before, the oxygen is a rad electron acceptor. It is. And then after that's all depleted, you go into your possible anaerobic environment, which still has nitrate, it has oxygen attached to in it. So it has to break this down first to get the oxygen out of it. That's why once you go into your fermentations and your anaerobic type of situations, it's just, that's how they get oxygen. Yeah, there's actual zones into it, but this is getting into a bit more environmentally. I want to keep the cannabis topic alive, I think. Fundamental reactions to the carbon cycle. You can see up here that electronics that are no, I'm not going to go into that. General trends. All different types of microbes from their environment. That's all their names. What's this? It's methane, I think. Oh, that's how it breaks it down. Okay, from the radox. Definitely not going there. Sedimentation. This is pretty fascinating. That inside of, if you've got a soil particle or substrate particle, more so soil because it's more dense, where if you've got peat or other things like that, it's not real dense. But inside of a soil, more so, because if it's 12 millimeters across, half an inch, inside of that could have zero oxygen, meaning it could be starting to reproduce anaerobic microbes or pathogens. So if you ever see something that's that big in your substrate, just squeeze it and break it apart so I can get oxygen in, so it won't have, because these stratified layers show the oxygen content. So clay especially, if you have a very heavy clay area and it's wet, there's a high chance that will be in effect. It's pretty cool. See before, remember this, you've got to be in favourable conditions for the spores to replicate and to germinate, and these are its favourable conditions if you want to go right into them. I don't. So like to summarise, like mechanisms of plant growth promoting rhizomes, rhizobacteria. So they do a lot of benefits. Like you're saying, where do you get them? You buy these ones. Oh, you can get them from other plants. So if you put that plant material, it'll be transmitted into its, if you put that plant material into a compost system, it'll be composted, and all those microbes and stuff will be mixed into the blend, and you'll have them eventually. This goes a bit too detailed. Cytophores, the iron chelators, hormones. Yeah, a lot of microbes produce hormones, and by having a microbe diversity, there's a high chance you're going to get a good hormone diversity as well. Like gibberellins is from gibberellic acid is from gibberellins, which comes from a microbe from them in Japan. They discovered it, fusicon or something, gibberellin. But that's a microbe that can produce hormones, as an example. Phosphate solubilization, went through that before. That's not in cannabis. That's in the legumes. That forms the rhizobium symbiosis. And that's the same one. Non-symbiotic nitrogen fixation is what we want with the azos down the bottom. Cyanobacteria like Anabana, or also Asperospiraplatensis. I've done a lot of experiments with that one. Yeah, non-symbiotic. Because these ones just work without anything else. So you know they're always just going to bloom and work. You need nitrogenase to break down nitrogen. Very good mechanisms come on those microbes. Other signals. This is what the microbes help and enhance and can do. They can produce all these things, can give the plant a bit of a break and help it with its processes. Phytohormones. Oh, I was saying this before. Plant produces the microbes produce phytohormones for the plants. Plant roots and plant growth promoting rhizobacteria interaction. Okay. Because, yeah, the microbes in the soil, they will go, this is the rhizophagy cycle, where they go out through the root tip, through the root hairs, and then back in through the root tip. It's a little cycle of heaps of different microbes that do that. Plant growth promoting bacteria. It's so awesome. How you can just get the right bacteria and fungi and microbes to run everything. Oh, that's just the different areas. See the roots inside, or the whole thing. The rhizoplane is touching the outside of the root, and the rhizosphere is this little area around the root. And it's a tiny area too. And this is probably where I should have started for the whole thing. But yeah, a gram of microbe-rich soil has up to 10 billion with a B cell. And a prokaryote, up to it also has good, healthy soil, has the same amount of viruses and pathogens in it. Believe it or not. Amazing. I couldn't believe it when I first learnt that. And the plant wants to have, because the plant has an immune system like us humans. So as long as everything's in check, it'll work out all right. That's why when people spray it, they'll spray out all the beneficials, and a lot of times these bad things survive. So they're spraying again, different type of spray, different type of this. So it's a battling cycle. Just go with organics, mate. Plant and microbe association. So there's three happen: harmful, beneficial, and neutral. So you neutral, commercials, beneficials is adding things and your plant growth promoters. Harmful is promoting all the pathogens, which is those similar processes I just described. Or you can inoculate your genetics, which I've gone and done this. Yeah, not going to go into this pathogen area. Just to inoculate, same way we get our jabs at the doctors to inoculate us, you do the same with the plant. So you administer little tiny bits on it. IAR, innate acquired resistance. So systemic, it goes through it. Innate is added to it. There's the quorum sensing bit. This is how I've used one of my substrates, was microbe worm castings, and then organic compost, and then perlite, and then here is manure. And this one I chose room manure, being organic. And the percents were 50% perlite, 20% organic compost, 20% vermicastings, and 10% manure. And that was ample for the whole grow. You might see a deficiency, so you might want to go and do a foliar spray. This is a rad experiment I did over a month. This is so that my phases of the microbial growth populations. So I did it with root tea, and I did it with different blends. I made a video on this. So it starts, and then 24 hours later, it's this amount. You can go up or down or stay neutral. Most of the time, they'll go through different phases. They'll start and they'll log when they eat up everything in their environment. Then it'll form a stationary plateau. Then everything's eaten and stuff, so they'll start and die off. That's where their spores will remain. See, that was just doing a fair bit of thing. So look at the root tea, because it's got lots of microbes in it. Start off at 1400, goes up to, no, it starts at 1000, goes up to 1300, still climbing at the end with only oxygen added. So that's pretty cool. After 2 days, it still wants to go up. So it's that's rare, because as you can see over here, most of the time, 12 hours, the microbes eat all their sources around it in a few hours. So it's yeah, it's cool. That's the difference. Microbes. Oh, this is what I was talking about before. The most common endophytes harbored in different tissues in the cannabis plant, obtained from different geological locations around the planet. So the red being fungi and the blue being bacteria. all these different areas on the plant have stacks. So if you grow like that, well done to you. You're letting the plant do its natural thing, and it will give you benefits back. You might get as high as a yield as the other folk, but you'll get a very high medicinally active plant, probably higher than the other folk. Is at the end of the slides. Okay, that'll do. I haven't seen any real questions actually. Yellow, blinky, Jono, if I decide to, oh, here we go, it's a bit of a question. Jono Who Cares says, if I decided to do a closed loop system and not run to waste with my feed, but recycle the warranty, would this work or kill my plants? It will work, mate, but you got to keep the plants will eat different bits in it, nutrient elements, so they might want to be replaced. You might want to monitor that so you're getting good with looking for leaf deficiencies. So for instance, nitrogen might be one of the first ones. So running your warranty or your hydroponic in your hydroponic solution is a rad way for organic hydroponics. Excellent suggestion. You must have seen that slide that I showed a while ago. And eventually it'll just deplete the system, mate, because they usually, when you do hydroponics like that, you might change the nutrient solution every week or every 2 weeks at the most. If you're doing in circulating system, not run to waste. So yeah, it should work pretty sweet, mate. And then you'll notice your deficiency. So once you get a deficiency in, you'll see it in the leaves, and then you can do a foliar application, which can eliminate that at 50 ppm. So you get that element, nitrogen. So you might get some nitrate, put them in some deionized water, make the ppm up to 50 or 100, sorry, and then you'll know that tissue, when you spray it on it, it's going to get that amount of the element. And you'll see within a few hours, 6 hours, it'll have absorbed into it and helped. Nitrogen being a very mobile element, it will absorb pretty quickly. It's the three immobile elements is calcium, boron, and manganese. So you can't foliar them and expect for a deficiency to improve. But yeah, good work, mate. I see Mace has hopped in the room. G'day, Mace. Nice to see you, buddy. GMF says, it will work. Jono, he's answering your question, I reckon, because he likes to help out too. It will work, Jono, but you may need to dilute things with water if ppm is too high. Leachette's high, yes. Yeah, so you want to put in your medium of, say, you might want to run it, dilute it down to, if you weren't tea leachette, maybe 700. Keep it at that. And when you go into flower, you might want to go up to 1400, 1300, cultivar dependent. And that should work pretty sweet. Running your worm tea and your hydroponic solution. Organics all round. And then, yeah, you could see which cultivars you work really well, and you could maybe focus on them in your solutions, because there'd be some that wouldn't like it. They'll want more this and more that. They'll whinge because they'll be fussy. But that'll work sweet, mate. And then you've adopted already a lot of microbes from your warranties, because it's come out, it's such a high concentrated amount. And the phosphorus amount in DNA is where it's sourced, where the plant gets it. So it has phosphorus solubilizing bacteria in the substrates. They'll go and see microbes, they'll consume them and release and make the phosphorus into the H2PO4-, which is the element that the cannabis plant uses for phosphorus. So that's how that gets into the plant. So the source of the microbes helps massively with phosphorus. So it's this living soil thing is really, really cool once you can get into it and make the system work for you. And then you'll get a lot of those, like the last one. Okay, put that last slide. Like this one, you'll get all of these naturally nearly there, because they'll be in your substrates, which will get into your plant material, and it'll be absorbed into that. So all of these ones in here, the endophytes, will be in all different parts of your plant tissue, because it'll be sort of circulated around. Then a little bit of deficiencies or sprays. I like to do a few sprays every now and then, but that's with beneficials. So especially going into flower, you definitely want to do a spray. And that's with no synthetics, no pesticides. It's only biopesticides and biostimulants is what I'd be using. So it's all organics. So and then that's going to protect it. So you definitely want to put subtilis on your spray. So day you've just flipped outside, or you've noticed you're in day 8 because pistils have appeared in your outdoor grow. So you might want to do a spray right then, because I don't spray after day 20, 25, because there's too much bud going on. And there's a chance that that spray is going to get in there and sit in there as moisture, and then that moisture is not going to be evaporated. So once the moisture stays there, no oxygen's around, bang, you're producing botrytis problems, because it loves it. It's all around everywhere. You just got to give it its environment for it to germinate and proliferate in, which is high moisture. That's why outdoor grows that are around rainy times in bud season, end of bud season, will always have this problem. Or if you're growing in an environment like Pacific Northwest, when I lived there on Vancouver Island in Canada, they've developed a strain on Lasqueti Island called Time Warp. It's Time Wreck. And that's to finish early, because in Canada there, on that island, you get a lot of moisture coming in, start of the wet season, and if you've got any outdoor grows in, they've got mold unless you pick it early. So by putting in Time Wreck, you can go, and it finishes naturally early, and like a month early in full sun. You don't have to do screening techniques or anything like that, shading techniques to try and get it to finish early. So it's pretty cool. Microbes make them work for you. They'll be working every part of the plant to only help you. They'll help protect, because they'll be occupying spaces. They'll be making mineral nutrients for the plant of its 17 elements it requires, and a couple of beneficials. Train wreck, not time wreck. Thank you, Lene. Train wreck. Yes, that was developed. See why slides are handy? Because they don't allow for errors like that. But it's cool to have cool people in chat that can pick me up on the things to help me with. It's good stuff. But yeah, if you've got things like that, that's one of the genetics from Lasqueti Island. Okay, what else? It has been an hour. Is there any more microbe stuff I can elaborate on? I love it. I'll show you the micro courses. They're fascinating, and I still look for more to come up to. These are my courses. That biomimicry was pretty cool. That means to fix a human problem, you look at what Mother Nature does. Like, well, I'm only going to give one example, because this is a medical cannabis show. I'm not going to get off topic. But on sharks' skin, they don't get bacteria. Why is this? Once they looked into it, they found that they've got little slits and cuts in it that are the size of nano. So milli, micro, nano, 3 times 3. So it's 9 zeros, decimal places down. That they found, that's how small it is. That you need a microscope to see it, but the bacteria can't colonize in these little pores. So what they did in hospital systems, they made door handles and things like that are with nano slits in them, that they don't have to clean anymore. So it's, yeah. Anyway, what's the other ones? Cell culture had a little bit in it. Food microbiology was fascinating. That taught me a lot about the moisture content of what microbes germinate in. Food microbial and food safety. That's right. That was better than the other one. Okay, what's the other few? No. Soil fertilizer. Uh-oh. Where's the microbe? Oh, microbial physiology and metabolism. Where's the first one that I did? Oh, applied environmental microbiology. That one. That's so rad. What's these interesting things? Oh, that's because of that disease. But yeah, this is a really, really fascinating course. Don't know if I should. Oh, that's the different one. Put yogurt in milk. I don't even know if you want me to go into this actually. Yes. Earth is a microbial planet. Microbes everywhere. Learn to live with them and use them to your advantage. No. Cell structure and activities. What's all this stuff? Flowering plants, cell structure. Oh, that's the, okay. That's the cell structure. Okay, good. This is getting right into it. This is some interesting stuff. The different areas that they can live in. So hyperthermophiles are the real hot ones. In the low ones, psychrophiles. How cool. In the Arctic glaciers, that's where you see bacteria on that, sea ice. But yeah, halophytes are ones that Jono's in Thailand might be there. He's got a halophyte because it's very salt tolerant. So there's all different names for different ones. Human gut, there's heaps. Cell numbers can be as high as, see that? 11 zeros afterwards per gram in the guts of microbes. Microbes, where do you get them from? Their environment. They're everywhere. That's how to do it. Sequencing, KR, 3D imaging, atomic force micro. Hey, this would have been a good one for me to pull out my microscope. Look at this protozoa. That's its face. Looks like a blooming thing out of, is it Star Trek or one of those scary movies? There's interesting microbes. That's coloured in too. They don't look like that. They look like that. These are different sizes. So the human eye can see down to, also here. My microscope goes to just in the nano range, and then all this other stuff you can't see. That flash technical ones. This is a bit more in detail to what we were showing before on the different types of shapes for their names. Cell structure and activities of microbes. Cytoplasmic, no. Where's some interesting things of microbes and cannabis? Oh yeah, this is, see how it's different organelles there, and this one here, the whole size of that is its endospore to reproduce. So that's why when the microbes die, big deal. They got half of them is made up. That's in most of them too. Not all of them have that endospore. There should be a photo in here of it. Population growth. That's what that experiment was that I did before. That's how it generally goes. And from my experiments to show how to continue it growing, you just had to do a few extra things to add to it. I've made some videos on that. How to continue the phase, the log phase. that actually the teacher for this course, that was her PhD. She continued growing phase for, I think, 9 months or something. So it can be done. Community growth. So you'll see a general, this is all round. Like if in your side of your compost pile, it'll generally go up and then down once the viability goes. And once you get to here, you know it's your harvesting stage. And you'll see that the pile will have shrunk at least half the size. That's a pretty good indication. And then you'll turn it up and it'll be black, beautiful geosmin smelling inside of it. No, we're not going to go into that. And look at all the chemistry and nutrition of the cell, of the microbes. So believe it or not, they need all these elements to survive on. And different ones need different elements. But in general, it's over here, 70 to 80% moisture. So that's why your compost pile has to be 60 to 80%, because you want to be cycling it. It has to have a lot of carbon and a fair bit of nitrogen. So you got them, pretty much a lot of microbes will proliferate and start to reproduce. And their endospores will germinate. It's getting technical. Litho means rock. Substrate, yeah, energy rich. This one. How they make their energy. Don't really want energy storage. Growth curve, done that a bit. Here's your hemocytometer. So this is how you can count your microbes. And I got one of these, and you don't pay this price. You just look online and you get a cheap one, which has under the microbes microscope. It's the squares don't really line up, and you can see why it's cheap. But I can't afford that price. But that's how to count them. Or you can get an automatic cell counter. Direct counting. So underneath the microscope, you'll see all these little squares, and that's how you'll count it. So this just goes into it with your hemocytometer. So to count them, you'll do your dilution first, and to see how many are left afterwards, and then that's the dilution rate. You'll put under the microscope, and you can see also what they are. So you go and say this much, and you put it under the microscope, make a slide of that, and you can easily see what's going on. Culture type. Yeah, not going into that much detail. Controlling growth. You don't want to know that, are you? They're the different ranges for the different. So as the compost goes into different temperature stages, so as more activity is being generated inside, heat goes up. It'll get into the thermophilic range, about the 65, 70, and then it'll start and break down all of the seeds and that type of thing that's bad for the pile. So that's how you get, you can put seeded material in, but just make sure you get it to the right temperature. You see it in wintertime, you might see steam coming off it. That's a good indication that the pile's nice and warm inside. No way we'll get into that stage. Classic model, aerobic, anaerobic, microbe respiration. That's pretty cool, isn't it? Oh, that's to do with its energy generated. We're not going to go into that bit. Chemostats, no. Proton motive force. This is the smallest motor ever in the planet, anywhere. It's inside of the microbes, and it's in their cell wall. So in their cell wall, they've got this little motor that generates to pump the proton motive force and generate their ATP. Just spin out. Here it is, this little thing. Smallest motor on the planet. Look, it's got a rotor and a stator as well. Isn't that amazing? It spins. That's what it looks like, but they've just made it looking nice for us. But yeah, it makes the energy for the cell. Prokaryotic. Nope. Cumulithotrophs, no. Looks like we might be there. Phototrophic bacteria. Cyanos. They're so good. No. Green sulfate, you can smell it. I don't even know what I'm showing you all these, actually. This might be the end. Yep, that's it. Morphological diversity, very good. That's how it reproduces. Magnetic bacteria, isn't that amazing? They got magnetic bacteria. They got bioluminescent bacteria. Sheathed. I wonder if there's another one this way. Stalk. No, it's not very interesting. Simpsons index. This might be all people for microbes to live. I talked about that already, but that's what they need if you want to actually reproduce them. Oxygen environment is very important. Talked about that before with the 12 millimeters across or half-inch across. Bioluminescence, rhizo deprivation, microbial mass. That's cool. Yeah, this is okay. That'll do. I think it's getting them moving a bit away from. That's pretty rad though. It's how they progress through time. Very good. Oh, it's now 20. Holy mackerel! Why haven't I seen all these comments? No worries. I was something, oddly enough, train wreck. It's a sativa dominant hybrid too. Yeah, it's a good cross though. Yeah, I didn't smoke it. Mexican cross Thai sativas and Afghan. Yes, microbes are fun. I reckon they are too. Very interesting things. So today's show was, ask a question now, because otherwise it's going to end. It's going to be retitled Compost and Microbes. And I only briefly talked about compost too. I could have went into detail, but that's that. So that's what I reckon. And yeah, it was good. Again, thank you for your ears listening, and appreciate the words that have been written in chat. It wasn't a great deal of questions this week. I hope there is some next week. Here's a question. Lene says, did you say didn't smoke train wreck? I think I tried it. Did I try it? No, I tried. There's a cross with it. It's time warp. No, I crossed train wreck with something wreck. Ice wreck. That one I tried. Is it ice wreck? It was so frosty. Ice wreck. The name doesn't ring a bell, but ice wreck. Anyway, it was a train wreck cross, and I think it was ice wreck, but I just tried it because it looked frosty under the microscope, so I thought, I'll try that one. And yeah, it wasn't anything too flash. And yeah, I probably wouldn't have tried train wreck knowing it was a sativa, because that's what I would have asked. Because yeah, I don't like the sativas. It's like as if I'm having heaps of cups of coffee or something. It's horrible. That's what you said you did. Okay, well, I might have, but I was probably thinking of ice wreck. I got the name wrong too. But yeah, today it's going to be changed. Today's show to composting and microbes. Yes, and if you've had any more questions, you're welcome to drop them underneath this chat show. That will be replayed because there's no more real questions coming through about it. And I hope you can bring your questions to next week's show so we can have some sort of a show. Because yeah, it helps a lot. Because now that I'm not making titles for the show, I don't know what to do. But I'd really like to help out and share the slides and stuff. So here's another comment. Lene says, thought you always raved about how much you liked train wreck back in the day. No, I liked how it grew and its technique for like their genetic modified that they did from their marker assisted selection. I like that. And it was grown pretty much because I lived, how close did I live? Only half an hour's drive from the barge that you used to get onto to go to that island. So I lived pretty close. So there's all sorts of crosses from that. And that was, that had been around for a while too, train wreck, for about 5 years, I think, when I was there in 2000. Geez, that's a long time ago. I left in 2018. I was there for 5 years. So that was already around for a while. Ice wreck. It was ice wreck. Yeah, I tried ice wreck. Yep, that's what it was. Because that comes more so to mind now. Yeah. All right. I'm not going to ramble anymore. Thank you again for listening. I appreciate your ears and everything else. And I hope that I can see you here next week. Happy growing, happy breeding, and good health to you all. Bye-bye.